This is part 4 of the Chapter 3 lecture notes, and this should go pretty quickly. There's just a couple more slides left. And we had left off on this slide, and I just wanted to make sure that you understood what I meant about the 1 to 2 to 1 ratio. So simple carbohydrates like glucose have a ratio of carbon, hydrogen, oxygen of 1 to 2 to 1. So there's twice as many hydrogens as there are carbons and oxygens. For example, I showed you the chemical formula for glucose is C6. So how many hydrogens would that be? If you double it, that would be H12. And since it's 1 to 2 to 1, the O would be just the same thing as the carbon. So C6H12O6. And another example, for example, ribose, which is a 5-carbon monosaccharide, is C5. And then see if you can do the rest of it. H and O would be what subscripts? So H would be 10 and O would be 5. Okay, pretty simple, but the difference is that lipids also contain carbon, hydrogen, and oxygen, but the ratio is not the same. Proteins contain carbon, hydrogen, oxygen, and nitrogen, and sulfur sometimes. And nucleic acids, DNA, contains carbon, hydrogen, oxygen, nitrogen, and phosphorus. There are two types of chemical reactions that tend to happen with these macromolecules. When the macromolecules are being formed, that is called dehydration synthesis. And I've shown you a couple of examples. This is an example down here of glucose plus glucose forming maltose plus water. So that's why it's called dehydration, is because a water molecule is removed from the reactants and water is given off as a product. So in other words, the reactants lose water. So dehydration synthesis is the way that polymers are formed. This is the synthesis reaction. If you reverse that, it's called hydrolysis. The way that polymers are broken down into monomers is called hydrolysis. So now we've got our maltose or our disaccharide that water is added to. One of them gets the OH and the other one gets the H. And that splits the maltose apart into its individual monosaccharides, which are glucose and glucose. So the breakdown reactions are hydrolysis, and the synthesis reactions or building reactions are dehydration synthesis. Okay, so this is one of the last pages, the second to the last page. Dehydration and hydrolysis reactions are catalyzed by enzymes. All the chemical reactions in our body are catalyzed by specific enzymes. Enzymes, remember, are proteins. They're always proteins. Dehydrations involve the formation of new bonds, and they require energy, whereas hydrolysis breaks chemical bonds, so that's going to be a reaction that releases energy. So dehydration reactions are what we call anabolic. And hydrolysis reactions are what we call catabolic. And breaking down macromolecules is what gives us energy, what gives our cells energy. Quick review: the structure and function of proteins. The monomers of proteins are amino acids. And then there are 4 levels of structure. Don't forget to review the primary, secondary, tertiary, and quaternary levels of structures. The different types of lipids, we learned oils, fats. Oils tend to be unsaturated, and fats tend to be solid and saturated. Oils tend to come from plants, whereas saturated fats or solid fats tend to come from animals. There are waxes, there are phospholipids found in our cell membranes. There are steroid hormones. The precursor of steroid hormones is cholesterol. Carbohydrates are where we get our quick energy from. The monomers of carbohydrates are monosaccharides, and nucleic acids are our genetic material. DNA and RNA are the molecules that determine our traits, that code for our traits. And remember to know the comparison between DNA and RNA. They have two different types of sugars. RNA has uracil, DNA has thymine, and then DNA is double-stranded, RNA is single-stranded. And macromolecules are large molecules. They're polymers. And the four classes are lipids, proteins, carbohydrates, and nucleic acids. They're right here. So let me know if you need any help. Good luck on the exams coming up after this chapter.